Have you ever grabbed, a sugar-free soda or maybe reached for a diet snack thinking, yep, this is the healthier choice?
Oh, absolutely. Lots of us do, right? Especially if you're trying to manage weight or keep an eye on blood sugar.
It seems like such a simple swap, an easy win. But what if one of those really popular sugar substitutes had a surprising kind of hidden effect? Something impacting, say, your brain?
That's quite a thought.
Well, today we're doing a deep dive into some, frankly, pretty compelling, maybe slightly unsettling, new research about erythritol.
Ah, yes, erythritol. It's a non-nutritive sweetener one you've probably had without even knowing it.
Exactly. We're unpacking a big study just published in the Journal of Applied Physiology, Barry and colleagues. Gives some really new ideas about how erythritol actually works on human brain cells.
And this builds on earlier stuff, right? Those findings that first sort of raised the alarm bells.
Precisely. So our mission today is to cut through the noise, understand what this new science is really telling us about erythritol in your brain, and, explore what it might mean for you.
Sounds good. Let's get into it.
Okay, let's unpack this then. For anyone listening who isn't quite sure, what exactly is erythritol?
Right. So erythritol is a sugar substitute. Technically, it's what we call a polyol or a sugar alcohol. It's a 4-carbon one.
Okay.
And it got the FDA's okay way back in 2001. Since then, its use has, it's really taken off.
Why so popular?
Mostly because it has a really minimal impact on blood glucose and insulin.
Right.
That makes it super attractive for people dealing with obesity, metabolic syndrome, or diabetes.
And it's super low calorie too, isn't it? That's a big part of the appeal.
Definitely. It's only about 0.2 calories per gram. Very low. And sweetness-wise, it's maybe 60 to 80% as sweet as regular sugar, sucrose.
Where do you find it typically?
Oh, it's in loads of things. Baked goods, candies, confectionery stuff, and like you said at the start, lots of artificially sweetened drink.
Interesting.
What's also interesting, though, is that it's not just an additive. It actually occurs naturally in small amounts, mind you, in some fruits, vegetables, and fermented foods.
Well, so it's in foods naturally and we add it.
Yes, and there's another layer. Our own bodies produce it, too.
Really? Oh.
Internally, from glucose and fructose, it happens in our red blood cells, liver, and kidneys. So the amount floating around in your blood is actually a mix of what you eat and what your body makes itself.
Wow. Okay, so that complicates things a bit when trying to study it, I imagine.
It absolutely does. It means understanding its total impact is, pretty critical and maybe trickier than we first thought.
So this is where this story gets a bit more complex, wasn't it? For ages, erythritol seemed great. Low-cal, diabetes-friendly. What could go wrong? But then you mentioned red flags started popping up.
That's right. For years, it was mostly seen as harmless. But then a couple of key studies really started to change the picture. One by Witkowski's group in 2023 and another by Lim et al. In 2024.
What did they find?
Well, the Witkowski study found a pretty significant association, and association is the key word here, between higher levels of erythritol in the blood and basically an increased tendency for blood to clot, what we call thrombotic potential.
Okay, more clotting. That sounds not good.
Not ideal, no. And they also linked it to a higher risk of major cardiovascular events, things like heart attacks and strokes.
Was that just in specific groups? Or more general?
That was the striking part. They saw this across different groups in the U.S. and Europe, both men and women, and this is important. It seemed to be independent of other known risk factors like high blood pressure or cholesterol.
Wow. So it wasn't just piggybacking on other problems.
It seemed to be its own factor, yeah. And Witkowski's team even did a small follow-up study. They gave people 30 grams of erythritol.
Which is like what? A typical amount in a diet drink?
Exactly, like a standard artificially sweetened beverage. And they found that just consuming that amount significantly boosted platelet reactivity, how sticky the platelets get, and the potential for clots, both in people and in lab tests.
Okay, that 30-gram number really brings it home.
It does. And then Lim's group, working independently, found something similar, a positive link between erythritol levels in the blood and a higher risk of heart disease and overall mortality, dying sooner.
So the evidence was definitely pointing towards something going on. But they didn't know how.
That was the gap. We had these strong associations, these correlations, but the mechanisms how erythritol might actually be causing these issues, especially its effect on the endothelial cells lining our blood vessels, That wasn't clear.
And those endothelial cells are really important for preventing strokes and heart disease, right?
Absolutely crucial. Endothelial dysfunction is a major starting point for those conditions. So we knew what might be happening, but we really needed to understand the how.
Which I guess brings us right back to this new study by Barry and colleagues. They tried to tackle that how question directly.
That's exactly what they set out to do. Their main goal was to figure out experimentally in the lab in vitro using cells what the direct effect of erythritol is on human cerebral microvascular endothelial cells. So, HCMECs for short.
Okay, hold on. Cerebral microvascular endothelial cells.
Mm-hmm.
So not just any blood vessel cells, but the ones specifically lining the tiniest blood vessels inside the brain.
Precisely. They were focusing right on the brain's own plumbing, those very delicate cells.
That's a really specific target. What were they looking for within those cells?
They zeroed in on three main things. First, signs of internal damage or stress, what we call oxidative stress.
Okay.
Second, they looked at the production of two really important molecules that control blood vessel tightness: nitric oxide, or NO, which relaxes vessels, and endothelin-1, ET1, which constricts them.
Right, the gas and the brake pedal for blood flow.
Good analogy, yes. And third, they measured the release of tissue-type plasminogen activator, TPA. That's the body's natural clot buster.
So stress, blood flow control, and clot busting. And what did they think erythritol would do? What was their hypothesis?
They predicted erythritol would basically make things worse across the board. Increase oxidative stress, mess up the NO and ET1 balance, specifically reducing NO and boosting ET1, and block the release of that helpful TPA.
And how did they mimic that real-world exposure, like the 30 grams in a drink?
They grew these brain endothelial cells in lab dishes and then exposed them for 24 hours to erythritol at a concentration of 6 millimolar. That specific concentration was chosen because, yes, it mirrors the blood levels you'd expect after drinking something with about 30 grams of erythritol.
So they really tried to make it relevant. Okay, the crucial part then. What did they actually find? Let's break down these discoveries. What was the first big takeaway?
Well, what's really striking is how fast erythritol seemed to kind of gum up the works inside the cells. Looking at oxidative stress first, they found that the production of those damaging molecules, reactive oxygen species, or ROS, was way up.
How much higher?
Significantly higher, about 75% higher in the cells treated with erythritol compared to the untreated ones.
75%. Wow.
Oh.
So basically creating a lot more internal static, more damage potential. Did the cells, try to fight back?
They did, yeah. The cells ramped up production of their own antioxidant defenses. Key proteins like SOD1 and catalase, think of them as the cell's internal fire extinguishers. Their levels went up too, SOD1 by about 45%, catalase by about 25%.
Okay, so the cells were trying to cope.
Few were trying, but here's the kicker. Even with those defenses boosted, it wasn't enough to counteract the big surge in ROS caused by the erythritol. The damage potential was still significantly elevated.
And why is that extra ROS bad, especially in these brain cells?
Excessive ROS can damage everything inside the cell DNA, vital organelles. And these brain microvascular endothelial cells are particularly sensitive. Too much oxidative stress can disrupt the blood-brain barrier, that crucial protective layer.
Okay, so increased stress inside the cells. Now, what about that blood flow regulation? You mentioned NO, the relaxer, and ET1, the constrictor. How did erythritol affect that balance?
It really threw it off kilter. This is fundamental for healthy brain blood flow. Nitric oxide, NO, is the good guy. Here it promotes vasodilation, relaxing the vessels for good flow.
Right.
They found NO production was significantly lower, about 20% lower in the brain cells treated with erythritol.
Less relaxation. So tighter vessels, potentially. Was it just making less of the enzyme that produces NO?
Interestingly, no. The total amount of the enzyme itself, enos, didn't really change much. It was more about how the enzyme was being activated.
So the machinery was there, but it wasn't switched on properly?
Exactly. Think of enos having an on switch and an off switch, controlled by specific chemical tags, phosphorylation sites. Erythritol seemed to flip the on switch down. Activating site phosphorylation was about 35% lower, and flip the off switch up. Inhibiting site phosphorylation was about 40% higher.
Wow. So a double whammy reducing NO production, even though the enzyme was present.
Precisely. Less NO means less vasodilation. Now, combine that with what happened to ET1, the brake pedal vasoconstrictor.
Let me guess, that went up.
You got it. ET1 production was markedly higher, about 30% higher in the erythritol-treated cells. And they also saw more of its precursor, big ET1.
So less relaxation from NO and more constriction from ET1. That sounds like a recipe for trouble with brain blood flow.
It absolutely is. It disrupts the whole balance, the homeostasis of these critical brain cells. It makes it harder for blood to flow properly, which, as you can imagine, is a really significant factor when we think about stroke risk.
Okay, so we have increased cell stress, messed up blood flow regulation. What about the third piece, dissolving clots?
Yeah.
That TPA molecule.
Right, TPA, the tissue-type plasminogen activator. This is your body's frontline defense against clots forming where they shouldn't. It dissolves fibrin, the stuff clots are made of, keeping vessels open.
Crucial for stroke prevention,
Yes.
obviously.
Absolutely essential. So they looked at TPA release from these brain cells. Now, the baseline day-to-day release wasn't really affected by erythritol,
Oh.
but, and this is a big but, the release of TPA in response to a trigger they use thrombin, which signals clotting is happening, that response was significantly blunted or weakened in the cells treated with erythritol.
Can you quantify that? How much weaker?
Well, the untreated cells, when stimulated with thrombin, nicely increased their TPA release by about 25%, like a healthy protective response. The erythritol-treated cells, they showed virtually no increase, flatlined.
Wow. So when danger signals appear, like a potential clot forming, the cells exposed to erythritol just couldn't mount that clot-busting defense properly.
That's what it looks like, yes. This impaired TPA release strongly suggests a reduced fibrinolytic capacity, a weaker ability to break down clots that's directly linked to increased stroke risk, and it might also affect the integrity of the blood-brain barrier itself.
Okay, let's pull this all together. What does this mean for us, for the listeners? This study seems to paint a pretty consistent picture. Erythritol hits these specific brain cells, increases stress, messes with the blood flow signals, and weakens clot breakdown.
It does paint a clear picture at the cellular level. And the big question, of course, is how do we translate these findings? Remember, they're from cells in a lab dish, in vitro. How do we connect that to actual human health?
Right. Petri dish isn't a person.
Exactly. But the significance is that all these changes, they observed the oxidative stress, the antinos issues, the ET1 increase, the blunted TPA release. These aren't random effects. These are known biological pathways that underlie problems with brain blood vessels, cerebrovascular dysfunction, and are known to contribute to a higher risk of ischemic stroke.
So the study provides a plausible how for what's seen in those earlier population studies.
Precisely. It offers strong mechanistic support. It helps connect the dots between seeing higher erythritol levels associated with strokes in large groups of people and what might actually be happening at the cellular level in the brain.
And you mentioned earlier, erythritol can actually get into the brain, right? It crosses the blood-brain barrier.
Yes, that's a critical piece. It's known that erythritol does cross the blood-brain barrier. This means it can directly interact with these cerebral, microvascular, endothelial cells in our own bodies. So these effects aren't just theoretical for brain health. The potential for direct interaction is there.
Okay, so strong cellular evidence supports the population data, and it can actually reach these cells in the brain. But like you said, it's still a lab study. What are the key limitations we need to keep front of mind?
Absolutely essential to remember. This was in vitro, lab cells. While HCMECs are a very good, well-validated model for studying brain vessel function, you simply cannot make definitive conclusions about clinical risk in humans based solely on this kind of study.
There's just more complexity in a whole living system.
Immensely more. Also, the exposure was acute, just 24 hours. We don't know from this study alone about the time course. Would the effects be worse with longer exposure? Would they fade if exposure stopped? We can't say for sure based on this work.
Which leads to the really big question, doesn't it? People often drink diet sodas or eat sweetened foods multiple times a day, every day. What about chronic exposure?
That's the million-dollar question, and the authors touch on it. They speculate reasonably that if people are consuming these things frequently, leading to chronically elevated circulating erythritol, then these negative effects on cells and tissues might become persistent, long-lasting problems.
But that needs more research, specifically in humans over time.
Definitely. The study itself concludes exactly that. Additional clinical studies are warranted on the potential long-term health effects of erythritol consumption. These findings provide a strong scientific rationale for doing those studies and doing them urgently.
Okay. So we've taken a real deep dive today into some pretty compelling new research. It shows how erythritol, this common sugar substitute, seems to negatively affect key brain cells in ways that are directly linked to a higher risk of ischemic stroke.
Yeah, specifically by boosting oxidative stress, disrupting those vital blood flow regulators, NO and ET1, and making it harder for the body to dissolve clots via TPA.
So for you, our listener, you're now definitely better informed about these, well, intriguing and maybe a bit concerning findings. The provocative thought we want to leave you with is this. When you're thinking about your choices for sweetening foods and drinks, especially things you might consume regularly long-term, how might these emerging insights influence your decisions? And what other questions does this raise for you about the hidden impacts of things we often just assume are fine in our diet?